Hello, thank you for viewing my video. I'm Chad, and for my topic, I chose medical genomics. Genomic medicine is an interesting topic to me. It is also known as precision medicine. It is this new medical discipline that you can use a person's genetic information to help them with their clinical care, help with their diagnostic and therapeutic ways of helping with diseases. And I have read up on the Human Genome Project, how it was able to completely print out our human genome in its entirety. It took about 13 years to complete, and it was actually completed earlier. We have our DNA technology to thank for that. And having the whole genome sequence is very beneficial. Now that we know that there are certain genes that can be linked with diseases, and we can easily identify them and try to do a great target on trying to develop new treatments. And again, we go back to why this is also called precision medicine, because it's almost entirely precise on what the disease you want to target and the kind of care that you can give. Now that with the Human Genome Project making it easy to know how the genome is sequenced, it's becoming part of routine clinical care that if you have some certain diseases, you want to have your genome sequenced, and it's even become popular with stuff like 23andMe and those DNA sequencing kits. So this figure from the textbook is one that relates to my topic. We have what is composing the human genome, and we learned in our class about the introns being the parts of the DNA being transcribed into RNA. These get spliced out of a growing RNA strand, and this little sliver is all of the protein coding genes that are important, but this only comprises like 1.5% of our entire genome. So the rest of it, either it has small function in the genome, and a bunch of it just doesn't even code for anything. And it's so strange how the 1.5% is the important part of our entirety. So this book, 1 in a Billion, it's about this child named Nick Volker who had an inflammation disease that caused holes to form in his small intestine. And it was the first ever kind of disease that any doctors had seen. And it was very difficult to try to treat Nick. He was a very young child when all this started happening. He was about 2 years old when he was first diagnosed with some problem. And he lived about 7 years of his life with having this horrible disease in and out of the hospital. And luckily, with the Human Genome Project able to sequence the human's genome, the one final solution to help Nick was to have his genes sequenced. And with that, doctors were able to figure out that it was just a single base pair error in his genome, and a bone marrow transplant was the thing that saved his life. This child is still alive, and it's amazing what genomic medicine was able to do in this time of need for a child. And this is actually my copy of the book. I took a picture of it, and I really enjoyed reading it, and that's kind of the reason why I wanted to do this topic, because it was very interesting to me how knowing what makes up a human is the reason why we are able to save more lives. And it's a really great read. I hope that maybe you get the chance to try it out. So, how genomics can relate to the RNA stability. We know in the central dogma that DNA goes to RNA, and then it goes into protein. So from DNA, our entirety, how do we look at the RNA that comes from it? The RNA that can come to code into protein is only about 1.5% of the genome. And some of the RNA that comes, it's more than just messenger RNA or tRNA or ribosomal RNA. There are these things called long non-coding RNAs. They have been identified in the human genome. There's not a full annotation of them, but they have been accounted for and seen in some human diseases that can help with some type of expression that's going on, an overexpression or an underexpression that can help figure out some kind of interaction that's going on with this type of expression deficiency or overexpression. These are some examples that have been documented with the long non-coding RNAs that NEAT1 is a type of long non-coding RNA. It's seen in ALS. There's a disc that we see in psychiatric disorders, and a long non-coding RNA that's starting to emerge. BC200 is seen in Alzheimer's disease. So with the genome sequence, we know what genes comprise the human genome. So we have a reference to look at if we want to study what kind of diseases are happening in a body, or what kind of tumors, what kind of gene expression is happening in tumors. So if we have a normal, that's our reference, thanks to the human genome. We use genomics in order to do a reference check on what's normal expression level and what's overexpression level or underexpression level. So the microarray, we have DNA probes that are used to tag certain genes. They're labeled with dyes, and the RT-PCR runs the DNA, so it's amplified, and we see gene expression levels. So the green would show normal expression levels, and if we start going into slightly over, we get a little bit of yellow, and greater overexpression can be seen in red. So a problem with having the genome just being our reference, only about 1.5% of the genome actually codes for something. But the other product, that doesn't really tell us much. And even with those genes, we know what those genes are. Not every gene happens to have a protein product. From DNA to RNA to protein, it's not always the case for one certain gene on the DNA. Sometimes it doesn't have anything. And sometimes that DNA just can be modified to have other types of products. So maybe there's something else that can be looked for. We know what the genome comprises of, but maybe that central dogma can help. If the ending product from DNA is protein, then what could be a different target for therapeutic purposes if we want a more direct target to mark in diseases? So what do we want to target more? We might want to target the proteins that actually causing some problem in a cell or in some of the diseases. So from genomics, we can go into another topic called proteomics. So the genome has the genes. From the DNA to RNA, we have our transcriptome, which is the entirety of transcripts, and then the proteome is the entirety of the proteins in the body. So looking at the proteins that comprise the body, they can actually be more useful in looking at the diseases where there's a misfolding of protein or one protein that's way out of order and there needs to be some kind of change that can happen. So using the proteome, we can identify the proteins that have diagnostic and prognostic markers to see what the disease, how it can progress, and we can use these proteins as ways to have therapies work. And so this are my references in case you don't believe me on my topic. You could read these references, and also I would really encourage you to read the book about Nick Volker. It's an amazing story, and it's amazing what we have come to in this day of advancing DNA technology. All right, that's my presentation. Thank you for listening, and what a semester we had.